Hello and welcome. I'm Luciano Lucas, and with me is Sean McElroy. He's a senior software engineer in the Aivia team and one of the lead engineers that has worked on the tools that you will see presented later on in this presentation. Together, we're going to introduce Aivia 9.8. This is the latest release. And this release is focused on the detection and segmentation of neurons as imaged by electron microscopy. Now, before we go into the actual presentation, just wanted to share what the agenda is going to be. We're going to start with a brief introduction to connectomics, including tools and software that is used in this field. Then some words about our motivation for joining this effort. And finally, the functionality that is actually included in Aivia and a few demonstrations at the end before we wrap up. So let's get going. Thank you for joining. I hope you enjoy this next 35 minutes or so. Neurosciences are definitely in the news these days in the broadest sense, and probably also because of the rise of artificial intelligence, which we have a few videos about and you can watch in our YouTube channel. There is a lot of hype, however. For example, science fiction about the possibility of downloading or uploading your thoughts into some sort of cloud system that then makes you essentially become immortal is something that newspapers like to talk about. We won't go into this today. It's an interesting philosophical topic, and it has serious consequences if it ever becomes possible or when it becomes possible. One of the groups working on this is the group led by Elon Musk. Again, a little bit still on the science fiction side, but they have made some progress. And the demos are actually pretty interesting from a conceptual point of view for now. But the brain-computer interface that they are working on is likely to continue to improve over time, and given enough time, they probably will get there. Now, on the real and serious side of this slide, so the bottom two pictures, there are very systematic efforts worldwide to map the brain's wiring, essentially map every dendrite, every synapse, and basically every connection that neurons make with each other. And the idea here is, and Jeff Lichtman summarizes it in these two statements, and by the way, I recommend his TED Talk, is really amazing. Not only is a great scientist, he's also a very good speaker. But he summarized in these two statements here. On the one hand, detecting and knowing the map of the brain, essentially where all the cells are, how they connect to each other, and understand the motives that describe these connections, can help in two ways. On the one hand, it is thought, and the hypothesis is that a healthy brain will have a certain type of wiring or a certain groups of motifs that are normal, and if the brain is diseased, or from a diseased person with a neurodegenerative disease, for example, then that wiring will look different. And if we can do that, we can map the normal wiring, we can try to understand and dissect out what the disease state looks like. On the other hand, understanding the wiring may also give us insight into how we learn and how memories are kept in our brain. So these are really broad questions, very interesting and very impactful to our humanity. And so this is certainly a very worthy cause to pursue. There are a number of outstanding research groups working on this topic already for quite some time, especially in the last 10 years with the advancement of particular microscopy techniques and also software that we'll mention in a moment. There is really quite a bit of progress, and it's actually inspiring to read the work that these leaders in this field have put out in the last few years. And this is not an exhaustive list, but it certainly is a great place to start if you're interested in this field and want to read up more. Please do search these. On the bottom here, I'm highlighting a few of my favorites because I'm more focused on the software side. So this is a technique, deep learning powered technique called flood filling networks. And that is pioneered by Viren Jain at Google, together with several other people, some of them on the list here, and they attempted and actually succeeded at automatically detecting a lot of neurons with very high accuracy. It's not only using deep learning, but then there's a lot of pre-processing steps to align the image, but also post-processing steps to aggregate the segmentation pieces that they get through the deep learning process. And aggregate them in an intelligent way to make sure that the segmentation is the highest quality possible. More recently, actually very, very recently, this is just now, 2021, a few weeks ago or a week ago, Sheridan and others have put a paper on bioarchives that expands or goes one step further on the flood filling networks and adding local shape descriptors as a restriction to how the segmentation happens. So locally, it looks for similarities in a few descriptors that are to do with the size of the object and the directionality of the object. And this makes the accuracy very similar to flood filling networks, but it's a lot faster than flood filling networks, which is one of the major criticisms of that technique. And one outstanding example of a collaborative effort and a team effort at the Geneva Research Campus is the FLY EM project. Perhaps the most important project currently out there in terms of already, the results are already out and you can already look at those results. It's truly impressive. There are, of course, several other amazing projects being undertaken right now and some outstanding results from other groups, but this is one of my favorites. So the imaging systems. There's essentially two types of imaging systems. One that is using typically a diamond knife, and in this type of system, essentially the sample is being physically cut with extremely sharp blade, and by doing that, you place it typically on a tape, and then you can image these very thin sections on this tape or on this wafer, and then reconstruct the serial sections. So this is one approach to this problem of imaging. The other approach is by milling with a high-powered beam, and essentially you're removing a very thin layer of the sample and then using a scanning electron microscope typically to image the surface that becomes visible. Once that is done, you do it again and then image again and so on. Each has its own advantages and disadvantages. Typically the block face type of imaging offers better Z resolution, typically isotropic, whereas the diamond knife type of approach doesn't offer the same kind of isotropic resolution, but it's easier to do it at high throughput. There are now hybrid approaches like the GC IB SEM, which is in fact a thicker sample, thicker slices are cut and then imaged with SEM as you would with a FibSem. Whether you use one or the other type of imaging modality for electron microscopy, the images will be enormous. It's typical that there will be several terabytes and even petabytes for the bigger studies that are that are happening now. So for this, you need dedicated software. Many of these projects that are ongoing have their own dedicated software, And here's a list of some of the software that is available. This is not an extensive list, but it's perhaps covers the most popular tools. And Berger in 2018 summarized all of these tools, pretty much all of them, and introduced a new one called VAST. And you can see there how it compares with everything else. So up to that point, that was perhaps from this comparison the most advanced and the most complete tool available at the time for annotation, visualization, and reconstruction of neurons. I've added here also Aivia for comparison on the same items that were proposed as good items to compare. And Aivia compares favorably on a lot of these parameters. There's only a couple of things that we don't yet do for now, but that we will work on in the future, and no doubt. One of them, and that's a short-term development, is to be able to work with online data sets. We already have this for our research projects to collaborate with our partners, and we'll soon bring that online for everybody to use that wants to use it in that way. The one other thing we don't have currently is multiple image stack of layers, so that this means if you have an electron microscopy image and a light microscopy image, we cannot show them together in the same field of view. But that's something we also plan to work on in the future. And finally, does this work with petabyte datasets? To our knowledge, it doesn't. We haven't tried it. We haven't actually had access to such a dataset yet. But once we do, we can certainly continue to work on Aivia to expand its capability to reach that kind of capability. Other than that, we do cover favorably pretty much everything else that is on the list here, and that's actually really encouraging. So what is our motivation? First of all, it seems like we have a tool that is at least does most, if not all, of the things that other tools do. And certainly as a matrix, it does very well, as you saw in the previous slide. But beyond that, we think that actually all of those tools are really well deployed within the research projects that they cater for. But perhaps they are not so easy for everybody else in the wider community to use. So not that many people in our view perhaps can access and efficiently and productively use those tools because they lack either the computing power or the human resources, meaning the technical know-how, how to use the softwares that are described there. And we also think that by enabling a wider community of researchers to contribute to this task of mapping the brain, we're actually going to help accelerate the rate of discovery in this field of connectomics. So it's with this motivation that we kind of worked on Aivia to add the functionality that we felt was needed in order to really cater for this kind of application. So Aivia, beyond everything I said, is in very active and dedicated development. We have a very good team, which I'll introduce at the end, in the acknowledgements, that works on Aivia regularly, and we have 2 or 3 releases per year, major ones. Also, it is packaged so that it can be deployed very easily. You don't have to install any other dependencies if you do it on Windows, and if you use a web browser, you don't have to install anything at all, not even the Aivia installer. Just access the website and you can start to work straight away. Of course, connect your data and start to work straight away. We do have a dedicated technical and application support team, and this is really a huge advantage to everybody that uses Aivia. And also importantly, it is possible to train your own deep learning models, either train from scratch with your own annotations, or you can start using the ones that we've already trained and then add a few annotations and use transfer learning to customize those models that already exist in our model zoo. in order to customize the model for your own data. So this is already possible today. In fact, it's already possible since Aivia 8 from 2 years ago. And Aivia does allow users to use Python to create their own Python scripts that run in parallel with Aivia, and so it extends its functionality beyond what we ship, of course. So what are we introducing today? And I'm primarily going to be focusing on the things that are new today. So that's the ones in the dark blue tiles. So the annotation, the tools for annotations have been either fully developed from scratch for 9.8, or they are significantly changed from the light microscopy version that we already had in the Neuron Composer, and we've changed them and augmented them so they work well for electron microscopy data. And so we can annotate both trees and outlines. So the neurons can have any shape not a cylinder, but any free shape. When we are doing the outlines, we have pre-trained the deep learning model so that you can quickly annotate using the annotation tool. It will predict the next and the next and the next slice deploying a pre-trained deep learning model in that region where you're drawing and trying to predict the next slice down. And it is possible, of course, to edit both the outlines and the trees, which is essential for proofreading and for obviously for correcting anything that was generated automatically. So the annotation part is essentially new today for electron microscopy. We also have a number of AI accelerated tools. We do ship a few deep learning models. I think there will be 3 when we release 3 deep learning models for the detection of neurons, so the neuron boundary, that were trained on different types of data, electron microscopy data, but slightly more contrast, less contrast, a little bit more resolution, a little more resolution. So there's a little bit of variety that you can pick from to get started. And then once you find one of the models that works reasonably well, you can further augment that model with your own annotations. We do have, and I mentioned that before, a system to train or augment your own models. And that's really important. We have that so that you can actually customize and get the best possible results. Excuse me. With 9.5, which was the previous release to this one, we introduced an initial tagging system. We've now expanded it so that it's even more functional, and it's catering for the functionality needed for electron microscopy. So you can have free text tags with comments, and even save presets, and as well as reload them, of course, and share them with a collaborator, for example. So these 3 tiles here summarize the functionality that is either brand new for 9.8 or has been significantly updated for 9.8. In addition to that, Aivia includes a wide range of other essential functions or essential kind of platform level specifications that are essential for electron microscopy work. So first of all, a visualization engine that is capable of rendering very large images in 3D interactively, and we do that. The data sets that we typically work with are around 1.5 teravoxels, and it's possible that it will support even bigger than that, but these are the types of data sets that we've worked with more recently. There are many advanced visualization modes, like Sean will show in a moment: ortho slicers, clipping planes, different hiding and showing of objects that are selected or groups. So there's plenty of ways to really dissect the area that you want to work in, and then focus on that area using those advanced modes for annotations. We also have, and I mentioned that we have this open and free deep learning model zoo. We started to populate it with Aivia 9.5 just a couple of months ago. There are 20 models there, and we'll continue to update that. In that model zoo, there are sample images, the trained models, and an explanation of what the model is for. That's very useful, and we'll continue to add that also as we develop more of them and we come across more data that we can use to develop more. And Aivia is deployed both on the desktop and on a web cloud environment. So that makes it possible to use it really anywhere and makes it easier to collaborate. So the two things that we believe are particularly useful beyond the things we've mentioned already and beyond the tools that are already in existence, like the ones I showed in the table at the beginning, is that you can do, of course, you can do the annotations, the visualization, the editing, all of this, but you can also edit and create your own deep learning models for segmentation. And this is something that sets Aivia apart. These models, over a long period of time, if we can get access to more of them, and the community has access to more of them, this will help everybody progress with their own work, because we can all learn from each other's efforts in training these models and potentially learning the best from each of the models to get the highest possible accuracy in a general application type model for electron microscopy, for the segmentation of neurons in electron microscopy. But this is only the functionality for electron microscopy and neuron reconstruction. Aivia actually has a lot more functionality that I won't go into detail right now because this is focused on Aivia 9.8. But since the release of the first Aivia in 2017, we've successfully added functionality. And I highlighted here all the ones that are related to machine or deep learning, the ones in white. So in every release, we had some functions that were kind of infrastructure and platform functions. Others were standard image processing functions like tracking or 3D object detection or neuron tracing, even in light microscopy data. But also we've been adding from the very beginning some machine learning capability. Initially was machine learning here for object classification, and we added 3 pre-trained machine learning models to classify neurons into different types of neurons. In Aivia 6, we actually introduced our first pre-trained deep learning model for the segmentation of neurons. It was our first attempt, and it was working, and actually several people are using it still. And we continue to add more and more functionality. Here we have the first pixel classifier. This is a random forest-based pixel classifier for image enhancement and image segmentation. Later on, we introduced Aivia 8 and Aivia Cloud, which is that platform, this system for updating deep learning models or creating your own that I mentioned earlier. And in the later releases, we not only accelerated the pixel classifier by making it GPU accelerated, but we also introduced this model zoo with 20 models for not just segmentation, but for restoration and for virtual staining. So that's really where we are now, and you can see in your own time, you can see that there's a lot of functionality there for the standard work that core facilities do. So if you're your core facility and you do electron microscopy as well as light microscopy, then Aivia can be used for both. And this slide just summarizes everything that is AI related that we've added over the years in the Aivia platform. So what does the architecture look like? How do we set things up to do the work within Aivia? So in this slide, I have separated the cloud on the top and local on the bottom, and we have Aivia itself. So the desktop application is down here. This application does all of the image processing that I mentioned in the previous slides. It does also the visualization. and we can do cell tracking, cell detection, neuron tracing. All of those applications run here. If I want to train a deep learning model from scratch or update one of the existing ones, I have a couple of options. One is to push that job to Aivia Cloud on GCP, which is ready to use for any Aivia user. And that runs, of course, on a very fast cluster of GPUs. And that's the fastest, most efficient way to train a model or to apply a model. But if you don't want to use that because either cost or maybe security reasons, you don't want to upload the data, or perhaps you just want to do it on a small amount of data locally, then you could run Aivia Cloud, the same functionality for training and applying models. You can run it locally using just your regular Aivia, and it uses whatever GPU or GPUs you have on your local machine. Now, another deployment we have, apart from this desktop deployment, is Aivia Web. So Aivia Web has all of the functionality. It looks actually exactly the same as Aivia itself, but it's running on a web browser. It's one running on AWS. And from there, from this Aivia Web deployment, you can do all the tasks that you would do on the regular Aivia and on Aivia Cloud. So you can train and apply deep learning models, you can do pixel classifier, you can do segmentation. We're using any of the 20 or so recipes that we now have for 2D and 3D image analysis. Oh, I almost forgot. We also have Aivia Community, which is a freeware, and I'll mention that in a bit more detail in this slide. So the Aivia product family includes Aivia itself, Aivia Cloud, Aivia Web, and the Aivia Community, which is a freeware used to visualize and annotate data. So it's ideal if you want to collaborate with several people. You can give them a link, they can just download. It's actually on our website, free download. Download the software, install it, load your data or your crop that you're responsible for, and then you can annotate that and save those annotations and send it to somebody, send those annotations to somebody that has the full Aivia, and they can then either train a model or update the model using the annotations that you created. So it's meant to be a collaborative tool. It's also a great way to visualize data and create beautiful video renderings of the data. In addition to the family itself, we then have what we are calling the friends. We have our GitHub repository where we put several plugins that written in Python for Aivia. We have the Aivia forum and as well the Aivia deep learning model library. I think I mentioned already all of these. I'll just open one of these here. So that's the model library, and you can see here the ones that we're actually showing today are these here, the ones on these three on the top. If you select one of these, it will then show you a sample crop of the image, the segmentation, a brief description. You can download the models and some explanation on how to use the models within Aivia. The GitHub repository is here. Probably loaded. Okay, here it is. And in here you will find the Python scripts that I mentioned, as well as the code related to a recent paper that we put out, which is called Arcan. It's a 3D Arcan. This is for denoising and increasing the resolution of images. Yeah, recommend that one for further reading later on. So there is a product family that covers pretty much all the needs of light microscopy, and now we've expanded the functionality also to electron microscopy. And even though we're focusing on neurons, really it's an annotation tool, generic annotation tool. You can use it for other things that are not neurons. But today we're going to be focusing primarily on the neurosciences. So in terms of workflow, the raw images come in and they can go through any of these paths. And even though this may look a little bit complicated, I'll break it down. The ones that are this like light blue means they are either machine learning or deep learning in nature. And you can see here the top is pre-processing, then detect is this layer, explore this layer, and output this layer. The E stands for enhancement, the R restoration, and S for segmentation. And the 3D tools don't sit really in a pipeline. You can access them anytime you want from any position you are in. So for the electron microscopy segmentation, we'd start with raw images. If we don't have any annotations and we just want to apply a model, we're going to go directly from here to the enhancement tools and then recipe console, run a recipe with a pre-trained model and get the results. Once we're here, we can use the 3D tools to edit the results, as you will see Sean do in a moment. If you don't want to apply the recipe console or the recipe that is created for you with a pre-trained model, you can start by creating annotations using the pixel classifier and getting some reasonable initial results, or you can go directly to the 3D tools, create all the annotations. This is what Sean will show as well. Create all the annotations manually, which give you some results straight away, and you can use those results to then train a deep learning model. That's summarized here as well. So the ground truth can be created in a number of ways. We have a recipe that automatically creates the neuron with a skeleton. You can do object segmentation, you can use a pixel classifier, or the contour editor, which is actually the tool that will be demonstrated today. So the contour editor together with the neuron composer create ground truth annotations for training a deep learning model. So that together with the raw data makes it possible to train a deep learning model. We use a variation of a 3D UNet as a starting point. And once we have that, then we can process a lot more data with the trained model or the trained models and get segmented, get results, which can be proofread in Aivia, so corrected, annotated further, and tagged for further analysis. Once we have proofread everything and we're happy with it, when it's appropriate, we can then use that to further augment the ground truth pool and generalize the model a little bit better. This is a process we call active learning, or it's part of a process we call active learning that we've actually presented recently at ASCB just last year. So downstream, those data sets look like this. I think you're an expert in this space perhaps, so you'll know what the electron microscope image will look like. The segmentation map in 2D will look like this, and then in 3D we can create nice renderings, mesh rendering of the neurons that are detected, and animate it for any purpose, for publication or just for a lab meeting. We do get a number of measurements out of these reconstructions related to morphology, shape, size, and frequency. So how many there are, how long are the segments, how thick they are, and a whole myriad of parameters related to the dendrites that we have detected. So what's on the roadmap, in case you are curious? So the next things we are working on already are the detection of synapses, vesicles, and circuits. Once we have the synapses, we already started on this. We also have already started on a collaborative version of Aivia Web that has all the data in the cloud. That's something we, as I mentioned, already have some of our collaborators using and will soon bring to everybody to use. We will be adding more editing options. We realize that it is complex. The task of neuron editing and proofreading is a complex task, and there are many edge cases that we are working through, but the current version already has a lot of those edge cases covered, but we know there are more, and we continue to add more solutions for those edge cases. So And also thank you for your feedback. As you start using it, we welcome your feedback so that we can quickly add those additional tools or scenarios that you want to cover. We do also want to add our alignment tools to Aivia. We already have alignment tools offline, and they work really well. We've presented those as well. And further down the line, we want to use both LM and EM to do segmentation. That's still in early stages in our roadmap, but it's something we have on there. And finally, and it might not be the last thing, it might come much earlier, we would like to start leveraging and working with multiple models and try to learn from multiple models. So models that each user creates. We don't need the data, but the deep learning model that is trained. We think we can get some information out of those deep learning models to create an even better deep learning model that generalizes across perhaps more varieties of electron microscopy data sets, which would again accelerate the rate of mapping, and that would be really great. Excuse me. Some of the work that we develop and work on during the year, some of it goes into Aivia itself and we release it as we're doing today, but not all of it. Sometimes we presented years ahead at ASCB or other conferences, and then only later it comes into Aivia. So here's a few examples. LM to EM, image alignment, is something that we did, I think it was 2018, no, 2019. 2019 we presented there at ASCB. Active learning was presented both 19 and 2020. And this is a process where we present the user with a result, and the user then annotates or corrects that result, and we learn from the process of correction. And so those edits go back into the training process so that we can retrain just the parts that are of lower quality, lower accuracy versus the ground truth, versus what the user, expert user, can define as correct. And we use those regions to retrain the model or to update the model. We've also already looked at and done neuron reconstruction using CLAM data, so where we have both LM and EM with some success, but that's still in relatively early stages, and we don't plan to have that in Aivia in the short term. We do plan, and this is one of the earliest posters we presented, was in 2018, in the reconstruction of the circuit based on connection points, so synapses, that we need to first detect. And we have a method for detecting the synapses already. So once we can bring that online, we can then also detect and study small circuits and then bigger circuits in the near future. So now I will pass it on to Sean. He's going to run through these 3 demonstrations. One that is running a fully automatic recipe through the recipe console, using one of the pre-trained deep learning models that I showed in the model library on our website. Then he will do one of the methods which includes manual editing or manual creation, creating the tree and then the outlines, and then the other way around. You'll start with the outlines and then create the tree. So that's the 3 demos that he's going to do. And so without further ado, I pass it over to Sean.
Hi, my name is Sean McElroy. I'm a senior software engineer here on the Aivia team, and I'm excited to show you 2 new features today. The basic features are going to cover EM image neuron analysis and detection. The first one will be a semi-automatic approach, and the second will be a more manual approach. So let's get right into it. So this is Aivia, and here on the left we have a great image from our collaborators at the Wang Lab. If I make it a little bit bigger, show off their hard work. It's pretty clean. Yep, so we'll use this image, and the first step in the semi-automatic approach is to use this new recipe, 3D Neuron Analysis EM. The old one has been renamed FL, no change on that one really. So since we have raw data, we'll need deep learning to convert it into a segmented mask fairly well. So we'll use this deep learning model that we've already got trained here. With our collaborators, we've worked pretty hard, and this is at least the third iteration, I don't remember exactly, of this model. So I'll just set some values. I actually already know these values. If you were doing this yourself, you'd want to use a region of interest, a very small region, and then you can try like low, medium, high and sort of tune it. That should be pretty fast, less than a minute to do. This one running right here will take over 20 minutes probably to run. So instead of doing that, I'll show you these results I already have prepared. And we'll set some calibration here. And so these are the results from the recipe. It'll output something like this. And one thing to note is they look a little funny because they're low LOD models. That's to keep the memory footprint down. I've got this option on. You can turn it off if you really want to force it. Although if it's unable to load all of it into your video card, it won't work. So you probably want to just leave this one on. When you get close, it'll go to high, and I'll show that now. Just pick one, and then we'll hide everything else and go close. I can show you the high detail here. Hopefully that comes across on the video. You'll notice it has a center line that you can see through the wireframe, or I can turn on topmost trace, maybe color it, like this. Yeah, and then I can show you what part of the image it was detected from here. And then pop this down so we can see it. So it's actually just on a few slices here on the top of the image. If you look here. So what we'll say is perhaps, yeah, this slice needs some small editing. So we want to improve the painted area here around this one. So we'll adjust the contour, the 2D contour, using the 3D Tools Mesh Contour Editor. So this is a mesh. We're going to adjust the contour. We'll edit the existing mesh after it's selected. I'll go ahead and turn off these options so you can see more clearly what I'm doing. So I'm just going to use the paintbrush and just paint something like this maybe. And then using 3 for the hotkey. It's also listed up here in the overlay, which you can adjust here in the overlay manipulator area. Just paint this and maybe paint this. Let's see, yeah, this one can use some painting, maybe something like that. I'm definitely not a good painter, but we'll say, you know, that's pretty good. Let's complete it with smoothing on, like that, and then just looking at this one by itself, there it is. So the last thing you might want to do after you adjusted any contours that you wanted to is you want to fix up the center lines. And so I actually like coin mode, and we're going to use the EM create mode. Oops, need a channel, and then like this. And we'll say we can use branch from point. So we'll just look at it real quick. So it's not very thick, it's just got a top and bottom. So starting from the top, say branch from the existing node, and we'll just place a few nodes over here. Build that one. And then we'll go down to the bottom one and branch from this one. I just placed some nodes like that. And then hide everything so you can see it. It's basically like that. Yeah, so that's basically all that there is to the semi-automatic approach. I'm gonna waltz straight into the manual approach, which is very similar. Using the same tools, you can actually just start making your own dendrite in EM data. As long as the orthoplane is on, you can just click on it and it'll make a node, and you can go down. In fact, I'm going to go down a few and click over here and go down a few more and click over here, go down, just sort of follow it. Oh, it's making a turn, so I'll just go back up over. We'll say that's, go a few more. Yeah, like that. And we'll say build that one. And actually I can just hide, so the orthoplane lets you configure these two separately. So you can do that. And then you can see how it goes behind the other one. So the other thing we might want to do, well, you're going to want to do if you're doing this manually, is you're going to want to actually paint the outlines. So we'll select it, and then go to the painting tool, and then go to the top because it's easier. And then I'm going to turn on flood fill to make this fast. And I guess normally you probably wouldn't use a mouse, or actually right now I'm using a ball mouse, a trackball, but maybe a pen or a tablet would be much better. But I'm just going to start going really fast here so we complete this quickly. And you'll note the flood fill actually closes itself off, so you don't actually have to do the last portion of it. We've got a lot to go, so we're going to go a little bit faster. Something like this. Oh, yeah, that's okay. We can make one more. It doesn't matter. So that's a good point. Anything you paint will now be associated to whatever center line, even if it doesn't exactly lie on the same z-plane or fit the same shape. It's up to you to draw as you wish. So go ahead and complete that with this one, smoothing on. We'll hide all these other things so we can see it. So there it is. And that would be a fully manual approach to creating a dendrite. So one last thing I'd like to do is show you, you can actually do it the other way. And I'll just do this real quick, but you don't have to start with the center line. You can actually start with the painted area. So if I just, so you noted I didn't edit existing, I started a new. You can also, yeah. And then I'll just sort of paint, and then paint this one, and this one, and this one, and this one. Probably those are attached, huh? And then maybe go down and paint some of this one. Oh, actually it belongs to this big long piece. But you get the general idea. So we do that, and then when we complete this one, you'll notice that it's in a similar situation. Where did it go? Oh, I should have added it to another object group. That would have been better. Yeah, it's a little tricky because they're all laying on top of each other. But here it is. And if I turn on the trace And actually, it's probably nicer to just do this. You can see, there it is. But our center line is bad. It's bad. So let's fix it. We'll go into EM Create and so start on this one. Coins. So here, and down, down. Oh, that's it. Okay, complete that. So now what we'll do is we'll actually get rid of these other points, which are kind of silly. And we'll just use delete, snip off this one, and then we'll go back to editing and do this. So now we can branch from this one again and go up. Up is the other way, sorry. Up. I think that's the end of that one. Turn off this and this one. And there is, oh, you know, it might be good to go off to the side there. So if we do that and that, we'll just branch from existing one, place a node way over there. And you know, you could even place one way down here for fun. And then turn off that. And there's our very poor center line. It's not too bad. So I hope you enjoyed that demonstration, maybe had a laugh or two, and hopefully you'll find these tools useful in the future. Thank you.
Okay. Thank you, Sean. Thank you very much for those demonstrations. Really appreciate it. And I hope it was useful for everybody to see a little bit how Aivia can be used to do manual and automatic segmentation, including some proofreading of the results that were created automatically. I would like to take this opportunity to thank everybody that has been collaborating with us to make Aivia stronger and better. This is the group that has been working with us in the last 4 years, specifically for the work that we presented today. Professor Rachel Wong and Dr. Wangqing Yu have worked with us over 2 years now, and we have a NIH funded project with them called ICA, Intelligent Connectomics Analysis Platform. And we continue to work on that, and it's a very exciting project that we are developing with them. So thank you very much to both of them and to all our other collaborators that we work on other parts of Aivia and even work that is not part of Aivia. Appreciate very much all your contributions. I'd also like to thank the Aivia team, in particular Sean McElroy for the presentation today and for all the effort in developing those tools that you saw being presented today. And there's even more that we didn't present today simply for time concerns. But we'll have another opportunity soon to give you a bigger taste of what Aivia 9.8 can do, and I'll mention a little bit more about that in a moment. So before moving on from this slide, I wanted also to thank Hideki Sasaki. He's responsible for a lot of the algorithms that we have in the Aivia deep learning pipeline, and Bryce Graft, who's also a senior engineer on the team, and he's been with us for nearly 9 years and has contributed greatly to how far we've gotten to until now with Aivia. So really, it's been is a great person to have on the team. There are many other contributions, and I appreciate everybody's effort. I'm very proud of everybody on the team for the effort they put in with every single release. Also, wanted to mention here the customer-facing team: Hoyen, Quinn, Patrice, Trevor. This is Doug, Doug Matthews, and Jim Palladino. These gentlemen and Quinn are the customer-facing team, and they're most likely the people that you would interact with, in addition to me, when you're trying to use Aivia and you have any concern or any question about how to use it. So in conclusion, I'd like to thank you first of all for attending the webinar, and just wanted to kind of summarize what I showed you today. So Aivia 9.8 offers a high performance image rendering and interaction tool for large EM datasets. That's the first that I hope you will remember when you go away from this presentation. Also, it supports annotations, editing, and proofreading, both of outlines and of the trees themselves, including the hierarchy and all the branching points and all of this that you saw also in the demonstration. It includes a wide range of AI-powered tools, some of them automatic, some of them for the semi-automatic detection of neurons. And it also enables you to customize your own deep learning models using Aivia Cloud. And finally, as you saw, we do collaborate to create these tools, and we listen carefully to our collaborators, in this case, on the neurosciences side. But in general, for all parts of Aivia, we have excellent collaborators and users that provide us feedback on a regular basis. And we take that feedback very seriously and try to either fix the bugs or create the functionality that is being requested of us. So thank you very much for that input. So in summary, this is what we are presenting today, Aivia 9.8. And I thank you for participating in this session. If you have questions, you're most welcome to ask them in a moment. I just wanted to mention before we go to the Q&A that we are running the AI Microscopy Symposia. This is a series of webinars. We're now going to the third session where we highlight both well-known and not so well-known researchers in the field of AI microscopy. So anybody that does microscopy and uses AI in their regular work, we are very interested in those. So this time around, we will have 3 outstanding speakers. You can go on our website and check it out right now. You can also search on Google AI Microscopy Symposia, and you'll find it. This symposia is coordinated between us at Aivia, the Aivia team, at the Aivia software, and with Leica Microsystems. So thank you also to Leica for their collaboration on this organization of this symposium. Soon, we are planning for mid to late February, we'll have Professor Rachel Wang and Dr. Wanqing Yu present their work in the field of connectomics, and especially in the field of studying the retina wiring. And I'm very much looking forward to seeing their presentation. We will be posting that on our social media and sending invitations to all of you in due course, so you can watch their presentation. I encourage you to try Aivia Community. It is a freeware, as I mentioned. It allows you to visualize and also annotate data. And if you want to get your own copy of Aivia, you can get a permanent license, or you can get an annual subscription of Aivia Web. So with that, I thank you for one last time. I hope this was useful. If you have any questions, you're very welcome to ask them now. You're also very welcome to go on the website and download Aivia Community or request your own demo license of Aivia right now. So thank you, and I'll pause there for the Q&A.